Push a muscle hard enough for long enough, and it will shut down. Total mechanical failure is the guaranteed biological outcome of sustained high-intensity training. In the short term, this exertion triggers an effect athletes refer to as the muscle pump. As the body demands more oxygen and nutrients to sustain movement, blood flow to the working tissue spikes, and local fluid pressure draws water directly into the muscle. This rapid fluid surge causes individual muscle cells to swell significantly. The result is a distinct feeling of tightness and a visible, temporary increase in the size of the muscle itself. But while the pump is a visible marker of effort, it happens right alongside an invisible decline. Eventually, the athlete hits a wall, and the muscle loses its ability to generate force entirely. To figure out why muscles actually fail, we have to look past the superficial swelling and examine the microscopic biochemical machinery driving the contraction itself. For decades, standard biology textbooks taught a specific explanation for this failure: lactic acid. The accepted model stated that intense exercise caused lactic acid to accumulate, creating a chemical burn that directly stopped muscle fibers from contracting. Modern physiological research reveals that this direct causal link between lactic acid and muscle fatigue is incomplete. Actual acute muscle failure is driven primarily by a localized ion shortage. Specifically, a depletion of calcium ions mechanically seizes the contractile system. To prove why the lactic acid model is flawed, we need to rebuild the exact molecular sequence of a muscle contraction from scratch. Understanding true fatigue means mapping the process of excitation-contraction coupling, tracing the initial electrical signal from the nervous system all the way down to the individual protein fibers. All voluntary muscle contraction is neurogenic. The sequence begins when an electrical action potential travels down a motor neuron and arrives at the neuromuscular junction. This electrical signal prompts synaptic vesicles to release a neurotransmitter, acetylcholine, directly into the synaptic cleft. Acetylcholine diffuses across the gap and binds to receptors on the muscle cell's outer membrane, the sarcolemma. This binding triggers a rapid influx of sodium ions, instantly reversing the membrane's electrical charge in a process called depolarization. This animated diagram shows the interior of a muscle cell. The wave of depolarization spreads across the sarcolemma and plunges downward into the cell's interior through a network of channels known as transverse tubules, or T-tubules. Deep inside the cell, the electrical shift hits voltage-gated sensors called dihydropyridine receptors, or DHPRRs, lining the T-tubule walls. These DHPR sensors undergo a physical shape change, interacting directly with adjacent ryanodine receptors, the RYR1 channels, located on a structure called the sarcoplasmic reticulum, causing them to open. This open channel allows a massive flood of stored calcium ions to rush out into the intracellular fluid. This rapid conversion of an electrical nerve impulse into a localized chemical surge provides the necessary signal for the next stage of the contraction sequence. With the cell flooded with calcium, the process shifts down to the sarcomere, the microscopic unit where the mechanical generation of force happens. The sarcomere is built from two interacting sets of protein fibers, stationary thick filaments made of myosin and sliding thin filaments made of actin. At rest, these filaments cannot interact. A protein complex made of troponin and tropomyosin acts as a physical barrier, constantly wrapping around the actin filament and covering its specific binding sites. This diagram shows the actin filament at the molecular level. The newly released calcium ions bind directly to the troponin C molecule, initiating a structural transition. The calcium binding forces the troponin to change shape, which physically pulls the troponisin strand away from its resting position. This shift fully exposes the docking ports on the actin filament. Without an adequate surge of calcium to move that physical barrier, the muscle's mechanical machinery remains permanently obstructed, making force generation impossible. The exact mechanism of how these proteins move past each other was independently discovered in 1954 by research teams at elite institutions like MIT. It begins the moment the actin binding sites are exposed. A single myosin head carrying stored chemical energy in the form of ADP and inorganic phosphate attaches to the actin filament to form a crossbridge. Looking at this schematic of the crossbridge cycle, we see the power stroke. The attached myosin head releases the inorganic phosphate and swings forward, generating exactly 2 piconewtons of pulling force. Millions of these microscopic power strokes occurring simultaneously pull the outer boundaries of the sarcomeres closer together, visibly shortening the entire muscle. After the stroke, the myosin head remains in a rigid, attached state. It stays locked to the actin filament until a brand new molecule of ATP binds to it, causing the myosin to release its grip. The system then resets. The new ATP molecule is hydrolyzed, broken down into ADP and phosphate, transferring its energy to move the myosin head back to its starting position, ready for the next cycle. Muscle contraction is a relentless cycle. To keep running, it requires an uninterrupted supply chain of both calcium to keep the actin accessible and ATP to continuously reset the myosin heads. Apply extreme, high-intensity anaerobic load to this system, like a maximum effort sprint relying on fast-twitch muscle fibers, and that biological supply chain begins to collapse. Under this heavy load, local oxygen availability plummets. To maintain force, the body relies on rapid glycolysis to synthesize ATP, a metabolic pathway that produces lactic acid as a byproduct. As the athlete maintains maximum output, the local reserves of readily available ATP, glucose, and glycogen are burned through rapidly. This data visualization illustrates the primary cause of immediate muscle fatigue. The timeline on the left tracks falling calcium concentrations. As the muscle exhausts its energy stores, it fails to release enough calcium to maintain the activation on the right. Without sufficient calcium ions binding to the troponin C molecules, the tropomyosin strands slide back into their resting positions, covering the docking ports. With the binding sites blocked, the myosin heads are physically obstructed from attaching to the actin filament. Force output instantly plummets. Lactic acid plays an indirect role here. Its rapid accumulation creates a highly acidic intracellular environment, which inhibits the biochemical pathways required to produce ATP. This ATP starvation means any myosin heads that are already attached to the actin cannot access the chemical energy required to detach, essentially jamming the microscopic gears of the cross-bridge cycle. Muscle failure is the mechanical seizing of excitation-contraction coupling caused by a total breakdown in the supply of calcium and ATP. The moment the intense exertion stops, the body enters a state called excess post-exercise oxygen consumption, commonly referred to as oxygen debt. Heart and breathing rates remain highly elevated, aggressively pumping reoxygenated red blood cells through the capillary network into the exhausted tissue. This massive influx of oxygen is utilized to clear out the accumulated lactate and begin synthesizing new ATP to replenish the critically depleted intracellular energy stores. With new ATP available, the biological cleanup begins. Specialized pumps, specifically the circopumps, consume this ATP to aggressively pull free-floating calcium back into the sarcoplasmic reticulum. EPOC is the body's vital mechanism for restoring systemic order. It balances circulatory hormones, resets local ionic gradients, and establishes the precise biochemical baseline required for any future physical movement. This entire process of exertion and fatigue exacts a heavy physical toll. Lowering a heavy weight under tension creates eccentric contractions, which cause microscopic tearing across the muscle fibers. These severe mechanical stresses and acute metabolic failures function as specific signals to the surrounding tissue. This sharp disruption in cellular homeostasis triggers downstream biochemical pathways, notably mTORC1, which command the cell to initiate immediate structural repair. Surrounding satellite cells activate in response, fusing directly to the damaged myofibers. This cellular fusion adds new myonuclei and contractile proteins, steadily increasing the muscle's overall size and maximum strength, a process defined as muscle hypertrophy. The localized breakdown of the sarcomere, driven by the specific depletion of calcium and ATP, functions as the precise signal required to initiate the production of new, stronger muscle tissue.